So we'll kick things off with our first keynote speaker, which will be Silvia De Santis. Silvia De Santis is a physicist by training. After finishing her PhD in diffusion MRI of complex systems at Sapienza University in Rome, she joined Professor Derek Jones at CUBRIC in Cardiff for a postdoc in white matter imaging. This was followed by Sir Henry Wellcome Fellowship in Tel Aviv with Professor Yaniv Assaf and Maastricht with Professor Allard Roebroek. And then thanks to a Marie Sklodowska-Curie grant, she moved to the Institute of Neuroscience in San Juan de Alicante, Spain, where she established her own laboratory, is now group leader and principal investigator on several projects which focus more on the characterization and validation of MRI biomarkers throughout the lifespan. Her talk today will focus on the use of neurotoxins to investigate the neurobiological substrates of several MRI biomarkers. The floor is yours, Silvia.
Great, thank you very much. Let me share the screen. Yeah, can you hear me okay? Yeah. So first of all, thank you very much to the organizer. I'm very excited to deliver my talk today at this exciting meeting. I guess the purpose when we apply diffusion-weighted MRI, the purpose is to try to model diffusion in tissue, in biological tissue, so that from the characteristic of the diffusion, we can learn something about the microstructure of the tissue. When we do this with diffusion tensor imaging, introduced more than 30 years ago, the aim is to then represent this information into spatial maps. For example, this is a map of fractional anisotropy in a rodent, in a mouse. But then in the last since the introduction of the TI, a lot of more complex and advanced models have been introduced with the aim of getting closer and closer to the actual neurobiological substrate. This is an example of axonal density and diameters, but actually the field is moving really fast. Anyway, what is done by obtaining these spatial maps is to get estimation of this information in cubes of tissue. These cubes are relatively small. They can go to from about 50 to 300 microns in rodents and a few millimeters in humans. But nonetheless, they contain a lot of different species, cellular species. This is an example of, I think, average composition in the cortex. This is gray matter. White matter will be slightly different, of course. So this is the neurobiological substrate that in its altogether determines what we see. And when we talk about validation, I like to think of providing a way of understanding the substrate and how this substrate defines the observed maps. Looking at the field of MRI, many studies never perform validation, but even when the validation is performed normally, my impression is that the sample size is normally very, very low, and this validation is mostly correlation based. Now, of course, not all the works, but in most of them, these are the characteristics. So this has driven my research in the last 4 or 5 years to try to introduce tissue models that could be capable to build a framework to validate MRI microstructure, MRI biomarkers. So the first part of this talk today, I will talk about this framework, which implies the use of neurotoxin capable of eliciting self-specific response with the purpose of validating the content of MRI signal. And then I will present a couple of applications of this, one more specific to gray matter and the other one more focused on white matter, and especially focusing on characterizing axonal pathology in the context of MS. And lastly, I will present what's another recent work from my laboratory that I call Validation When Validation is Not Available, which is an atlas of diffusion and another MRI contrast registered in the Allen space. Let's start with the first one. In the last 4 or 5 years, I've been working a lot with neurotoxin because what I find intriguing is that by controlling the window of activation and by combining different compounds and also pharmacology, is it possible to target specific cells and recreate and isolate condition of interest. What I'm going to talk about is particularly three kind of toxins: the lipopolysaccharide, in which there is a microglia and astrocyte activation following intracranial injection. I will also talk a little bit about some data obtained with injection of isolecithin, which you will probably very well know that is affecting myelin, but also generating some transient inflammation. And lastly, I will talk about injection of ibotenic acid, which will affect the neural body, which create neuronal loss, but also concur with inflammation. So in the first model, we are interacting mainly with the glia, microglia and astrocyte, challenging the reactivity. And it's possible, as you can see from the activation window, is it possible to isolate microglia during activation during the very first hours? And by combining the IPS model with administration of the compound PLX5622, which is an inhibitor of microglia growth, which effectively depletes up to 90% of microglia in the brain transiently, we isolate astrocyte activation. For the lysolecithin, injection of lysolecithin, we tried the injections in the gray matter. They cause reduction in the myelin content after 2 weeks, and transient inflammation, which is however resolved in this window of time. And lastly, another interesting paradigm is the injection of ibotenic acid. Why? Because in the injection around the injection site, it generates neuronal loss and also microglia activation with proliferation, which is something that we don't see with the lipopolysaccharide. And also it can be used as a model to generate axonal pathology in the white matter that is, containing the axons the axon sprouting from the neuronal bodies which are damaged by the ibotenic acid. So together we can we have generated 5 different conditions, one in which we isolate microglial activity through injection of lipopolysaccharide in the hippocampus, and by looking at the tissue, the MRI in tissue 8 hours post-injection, which is when astrocytes are not activated yet, while there is significant change, morphological change in the microglia, suggesting reactivity. The astrocyte reactivity instead can be achieved by injection of the same toxin, lipopolysaccharide, in the hippocampus, but in animals that are treated previously with the PLX5622, that, as I said, depletes almost completely microglia. The third condition is microglia reactivity and proliferation. We don't observe proliferation with injection of IPS, but we do with injection of ibotenic acid in the hippocampus by waiting about 2 weeks after the injection. Of course, we can also generate models of demyelination. This is more usual in the field. And then lastly, by looking at the white matter of the fimbria, which is the tract projecting out from the hippocampus in a model where the hippocampal neural body are damaged, we can generate axonal pathology, axonal damage, about, again, 15 days post-injection with no signs of inflammatory reaction. That is probably already resolved by then. We used, so the first application of this framework was in a paper that we published last year in the laboratory, where we used this to look at neuroinflammation, which is a really trending topic in the last few years, because of his double role in the homeostasis of brain, having both positive and negative functions, positive in to promote tissue modeling and repair, but also negative as in increasing neurodegeneration and also being associated with aging, for example. So the first thing that we did was to use LPS. I will skip a little bit further, to use injection of LPS in one hippocampus and saline in the other, different waiting time to generate the different models, and then proceed with the MRI acquisition and immunohistochemistry. Through immunohistochemistry, we are capable of reconstructing in 3D cells. This is immunohistochemistry with staining EBA1, which stains microglia. And as you can see very well, there is a changing morphology, a very clearly changing morphology from the control, in which the microglia has a highly ramified processes to processive retraction and increase in the cell body size after 24 hours post-injection. And this phenotype is reversible. If we wait 2 weeks after the injection, it's totally reversible. We can quantify different characteristics from these 3D reconstructions of the histological stainings. So by comparing the two hemispheres, it's clear that activation, reactivity of microglia is reflected in changing morphology of the reconstructive microglia, so that there is a clear and highly significant decrease in the process density, an increase in the cell size, and also change in the dispersion. By using multi-compartmental models for MRI, where we can isolate the water diffusion in processes, its dispersion, we also estimate cell body size in the restriction. We can compare the same parameters that we estimate with the histology by estimating it with the MRI, and we see highly remarkable patterns of decreasing this stick fraction, increase in the sphere radius, and also changes in the dispersion. What is interesting is that if you look at the gray, dark gray plot, it seems that this increase is specific of microglia, because when we measure the same quantities in animals that have had their microglia depleted through injection of the compound PLX 5622, then we don't see significant differences between two hemispheres. So these, we can of course visualize the different parameters voxel-wise, and in this case, this is visualize the process density is visualized in the two hippocampus when there is a clear decrease in the injected one. And we can also make a similar story for the astrocyte, where instead we look at the volume, the total volume of the astrocyte by doing a convex hull analysis. The same framework can be used to test other conditions. For example, more recently we used it, or part of this is also contained in the paper, but we use it to test sensitivity of conventional MRI to microglia reactivity to different conditions, microglia and astrocyte reactivity. And for example, we confirmed association between mean diffusivity and microglia reactivity that was already published in several papers, and also confirmed its non-specificity. Interestingly enough, we also confirmed the sensitivity of the ratio T1-T2 ratio, which is a proxy for myelin content, to lysolysozyme-induced demyelination. But interestingly enough, we found that this ratio is also sensitive to astrocyte reactivity, where myelin is untouched. And lastly, we found that T2 star is sensitive to microglia reactivity only when the microglia reactivity is accompanied by neuronal death, such as in the ibotenic acid injection, which is very possibly due to possible metal accumulation due to changes in neuron metabolism that is following neuronal toxicity. We can also use the same framework to test the sensitivity of MRI to microglia reactivity in vivo and ex vivo. We scanned also the brains before undergoing histology. They also undergo an ex vivo protocol, and we can see that there are comparable changes in the disease restrictive fraction, there are comparable changes in vivo and ex vivo. Perhaps there is a little bit a bigger effect size in vivo. So the sensitivity is overall preserved. So the conclusion of this part of the talk is that by using a combination of neurotoxin and pharmacological treatments, we can elicit focal cell-specific alteration, creating a framework for testing quantitative and validate quantitative MRI estimation. But also, we can test the sensitivity to the diffusion MRI to microglia reactivity, different conditions in vivo and ex vivo, and also other parameters. Let's move to the second part, in which we use a similar framework, but we focus more on white matter. In this case, the interest was specifically focused on multiple sclerosis, which is a chronic inflammatory demyelinating disease, whose main pathological hallmarks are the white matter lesions. Since a few years, my laboratory is in collaboration with other laboratories is interested in the degenerative aspect of the disease because of its clinical relevance. So axonal damage is believed to be more closely associated to permanent disability, for example, than the demyelinating aspect of the disease. So for this, it's of particular interest to investigate the early stages of the disease. We decided to use Excalibur, which is a modeling strategy introduced a few years ago now to estimate the axonal diameter from diffusion MRI. Under the hypothesis that acute axonal damage would manifest as an increase in axonal diameter. And this is due to, this is compatible with animal model and postmortem evidence in multiple sclerosis, and it can be either due to the preferential loss of small axons, but also to the blistering phenomenon, which seems to be an early phase associated to axonal damage. So in this part of the work, we wanted first to establish the framework for validation of the preclinical model or in the preclinical context of validation of sensitivity of MRI to axonal pathology. And we did this using the damage caused in the fimbria by injection in the hippocampus. And then we applied a similar framework in humans, in MS patients, to characterize the same quantity and highlight early alterations suggestive of axonal pathology. So again, ibotenic acid injection caused neuronal death in the dental gyrus and the degeneration of the fimbrial tract, which we confirmed through histology with two approaches. So the first of all, we confirmed the neuronal death in the dental gyrus, which is highly significant, by staining for neon-N, neuronal nuclei. We then looked at the fimbria projecting from the hippocampus, and we detected increased axoskeleton protein density, suggestive again of axonal damage. But then the final proof was through an electron microscopy STEM, an electron microscopy paradigm, in which we measured increased axonal diameter in the injected hemisphere, highly significant without alteration in myelin, a trend but no alteration in myelin, and no changes in the number of axons. So we now compared MRI-derived axonal diameter proxy in controlled and damaged fimbrias. And what we detected is a generalized increase in the MRI axonal diameter proxy in the hypotenic injected hemisphere. This is visualized in one subject with the fimbria projecting out of the hippocampus, and of course, if we quantify it across the wall, fimbria is significant. And interestingly enough, this change. The measured axonal diameter is significantly associated with the staining of neurofilament, the axoskeleton protein. So we move on to look at MRI data in humans. We had, well, it's a relatively small low N in this study, 11 subjects compared to 10 controls at the Connecton scanner in Boston. And when we compare the normal appearing white matter of this patient outside the lesion, so lesion are excluded from the analysis, we see a generalized increase in the axonal diameter. This was published already a few years ago, but the new finding is that if we look at the association between this increase in the axonal diameter and the disease duration, we found out that this increase is only significant for patients who have disease duration shorter than 5 years, suggesting or confirming our hypothesis that this is a marker of acute axonal damage and not a later stage of axonal damage. So to summarize, MRI-based axonal diameter mapping is sensitive to early stages of axonal degeneration, which is detected as an increase in the measured MRI axonal diameter proxy. And in humans, we uncovered diffuse axonal damage in a normal appearing white matter of MS patients, which is inversely associated with disease duration, confirming our hypothesis of axonal damage increase as an early sign of axonal pathology. While during the revision process, we had to add very interesting data on this, and I think this is something that is of interest to the community. So we performed the electron microscopy experiment, which is to confirm the increase in the axonal diameter in the damaged fimbria, which is kind of the gold standard for structural type validation. But I'm not sure of, at least I didn't have in mind the main limitation, which due to perfusion and fixation. So it is quite well known that there is a shrinkage factor, which is due to the error processing, and it has been very well characterized in literature to be around 15–16%, and it can be used to correct back the estimates of the structural size. But indeed, also the perfusion and fixation shrinks the brain. And when I looked at the literature for different fixatives, similar to the one that we are using for the electron microscopy processing, the shrink factor is surprisingly very big. Depending on the fixative, it can go up to 68%. So we decided to measure it ourselves. And we found that for our preparation, which involves 10 days in fixation, the total shrinkage was about 40%. So this is quite a relevant amount of shrinkage. Of course, it cannot be used, this factor, to correct back the estimates of the axonal diameter, because extra and intra-axonal space can be differentially shrinked. This is almost possibly the case. So perhaps cryo-EM can make things better, but anyway, we need to consider when taking electron microscopy as gold standard, we can consider the difficulties of drawing quantitative information. So if I have a couple of minutes more, I just want to show a last bit of work that we are doing in the laboratory, which is about moving our MRI from rat to mice, which are advantageous for several reasons: the possibility of genetic manipulation, the resources that are available in the field. So we wanted to extend the MRI framework, and we wanted to use to build for ourselves a tool for the analysis, which is the Atlas. We looked at literature. We couldn't find something that was useful for us, mostly because most of the atlases that are available do not include female. Sex differences in brain aging is one of the main themes of my laboratory, so we really wanted to have equitable representation. So we decided to build a template ourselves, and we did so by acquiring a large number of female and male animals, with which we constructed an atlas with anatomical and FA-MD contrast. And then when it came to parcellation, we wanted to include the Allen, the parcellation using the Allen, which is the most used resource in mouse analysis, which has a lot of data that can be explored. This is why I am talking about validation without validation. So to do so, we wanted to parcellate the brain. We did a parcellation according to the Allen brain. And to prove the utility of having the MRI and all the Allen Atlas resources in the same space, we explored the correlation of diffusion metric with cell count from transcriptomic data that is published and associated with the Allen Atlas. And for example, we find that mean diffusivity is correlated, significantly associated with and only with microglia count. It's not associated with neuronal count, oligodendrocyte, astrocyte, etc. And actually, when we prepared the graphical abstract, we saw that by choosing the proper scale, an MD map and this cell count from transcriptometry were looking incredibly similar. This is compatible with also recent finding of the laboratory. This paper of 3 years ago, when we looked at the paper was about the impact of alcohol on brain microstructure, but we used again, LPS, PLX to manipulate microglia, and every time we saw that the indifusivity was increased for this manipulation. And finally, if I have another minute, the last bit, we try to quantify the impact of using an atlas made with only male animals. And to do so, we calculated the formation of that the brains have to undergo to match a template built with female males and a template built with only males. So as you can see, the deformation when the atlas is built with only males are much bigger, are definitely bigger. And this generates spurious significant sex effect on new data that we have acquired longitudinally to test a general linear model with effect of sex and age. When you compare the analysis in which you use, everything is the same, but you compare the analysis in which you use a template done with male and female, like the one on the left, nothing, the sex term is never significant, but when you use a template for animal of both sexes made only with male, then we generate a bias introducing spurious significant sex effect in 5 regions belonging to both gray matter and white matter. So that's it. The conclusion of this part is that the feasibility of advanced MRI imaging to map glial activity in the mouse brain, we tested feasibility of advanced MRI to map glial activity in mouse brain, and we stress the importance of building standard resources, including animal of both sexes, because using male-only standard resources can result in spurious sex differences. And that's it. This is my team. Thank you very much. Thanks again for inviting me and you for your attention.
Thank you so much. That was super cool. Sajad, do we have any questions in the chat yet?
Yes, we have a few. So the first one is from Dorian Postino. What is the biological mechanism that causes thicker axons in MS? Is higher axonal diameter expected to happen in other neurodegenerative diseases?
That's a very interesting question. Can be related, so it's possibly a initial compensatory mechanism. In fact, one of the papers that I cited was showing that this is reversible up to the beating is up to the beating stage is reversible. Possibly compensatory mechanism, it's failing, the axonal transport is failing, maybe because of the interaction with the oligodendrocytes. There can be an accumulation of mitochondria, but I think it's still not known fully.
Thank you.
Yes, and also it's common to also other neurodegenerative diseases. Yes.
Another question is from Jin Cheng. How long before clinical MS onset the change in axonal diameter started to happen, and is significant enough to be seen with MRI?
Great question. So I have to say that these results are based on a very small sample size because of the the access to the scanner, the connectome scanner is not so easy. So they have to be replicated on a larger sample size. But from what I see, we could cast the segregation between high and low axonal diameter was optimal when we draw the line at 5 years. I'm not sure how earlier we can go from this. So again, we need a bigger sample size, but this is what our preliminary results indicate.
Thank you. We have another question from Olivier. By combining microstructural markers like MD, T1 over T2 or T2 star, could we better estimate inflammation?
Definitely. Multimodal, machine learning. Actually, if someone wants to I'm open to share the data to do all the possible trials. I think what is nice about this framework is that can be used to answer many questions. We made sure that we isolated as much as we could, because of course there will be other more subtle modification, but we made sure that we isolated to the best that we could microglia activity, astrocyte reactivity, etc. So this is a data set that can be challenged in many, many ways, and we are open to share the data.
Great, thank you. I have a couple more questions, but I think we can go on to the next one, and we can go back to them in the discussion session.
Okay, all right.
Okay,
Sounds
thank you.
Thanks again, Dr. De Santis.